record. All right. So in the last part of control and coordination chapter, we are going to look at control and coordination in plants. All right. So when we come into this topic, control and coordination, we are looking at the transmission of signal or message, right, from receptor to coordinator and coordinator to effector in mammals. Okay, the first section. Then the second section is we also want to look at the transmission of signal or messages in the plant for them to respond to stimuli, okay, for them to carry out necessary action, right? So we're going to look at 3 examples for plant. The first example they're going to look at is actually to describe the very rapid response of the Venus flytrap to stimulation of hairs on the lobes of the modified leaves. So these are the modified leaf. That means normal leaf looks like this, right, for photosynthesis. But for the Venus flytrap, the leaf has been specialized in their structure so that it's used to actually trap insects, okay? Just like flowers are actually a specialized leaf to form petals to attract insects, okay? So they're specialized leaf which form lobes, okay, in order to trap insects, and how the closure of the trap is being achieved. So we're going to look at this process, very interesting process of Venus flytrap. Okay, I'm going to skip all this. So today we're going to look at communication system in flowering plants. Okay, so communication in the system in flowering plants is very similar in mammals. We can use electrical communication, so the transmission of action potential along the membrane of the plant cell, just like along the neuron of mammals. And secondly, using chemical, right? The chemical in mammals, we use hormones, okay? And in plant, they use plant growth regulators. Now, plant hormone has got a official name, which is called the plant growth regulator. So this is a term used to denote plant hormone, plant growth regulators. So example of plant hormone or plant growth regulators you can look at are auxin and gibberellin, short form for auxin IAA, short form for gibberellin GA. Okay, but today we're going to focus on only the first one, which is the electrical way of communication to achieve a very rapid response to a stimulus. Okay. So flowering plant has communication systems which allow them to respond to any changes in the internal and external environment, just like us. Okay. So this communication system function is to transmit messages either by electrical or by chemical. So by electrical is if you want the response to be very fast, okay, we have to use electrical way of communication. So that one uses action potential. So very few plants will use this type of communication system because it is quite expensive, okay, because it requires the movement of ions across the membrane. It requires the formation of concentration gradient of ions across the membrane, which means you need the active transport, ATP. It requires the formation of the channel proteins. So synthesis of proteins needs energy. So it's more expensive. But if you want it fast, then you have to produce all these things. You have to use ATP to achieve this fast communication. So very few plants got it. This is for very rapid response. Example we're going to look at is the Venus flytrap. Okay, of course, mimosa also have got a fast response, right? You touch the mimosa leaf and quickly it will close, isn't it? Right? Chemical. So most plants communicate within themselves, okay, within the cells in the plant body using a chemical, which is plant growth regulator, right? So this plant hormone is called plant growth regulator, short form PGR. It can be natural plant growth regulator produced by the plant. It can be synthetic plant growth regulator produced by human to stimulate the plant to carry out certain response. Okay, like to stimulate the fruit to become, to the flower to grow to become fruit without fertilization. So what hormone do you use, okay, to stimulate germination of seeds without having, let's say, suitable condition, they can still germinate, okay? So this is a use of PGR. So PGR is plant growth regulator, is substances that affect plant growth and development. That's why it's called plant growth regulator. Regulate means to control when to grow, how much to grow, when not to grow, okay? Or when to develop, when not to develop. So this one is the Venus flytrap and this one is the mimosa. Okay, so you touch the leaf of mimosa, then very quickly it will close because they use electrical way of communication. Okay. Okay, let's look at the Venus flytrap, right? The structure first overall. So it is a carnivorous plant because it feeds on animal. What kind of animal? Small insects, okay? Small insects. Okay, why do they want to feed on small insects? Because they want to obtain nitrogen compound from the body of the insects by digesting them, okay? Because nitrogen is very important for plant to synthesize DNA, to synthesize amino acids. So it's very important compound. So they want to obtain this from animal because animal got a lot of proteins in them, okay? So therefore, they have got very specialized leaf to do this function only. So the structure of this leaf, it contains three special sensory hair or trigger hair. Can you see three here? One here, okay, two here, and three here. So every lobe, okay, this is called lobe, like your ear lobe, okay. So every lobe has got three hairs. So they call it three sensory hair because they are like sensory receptor, okay? Sensory hair or trigger hair. So this is where they can be triggered. This is where they receive the stimulus, okay? So every lobe got three sensory hair or trigger hair. And then the trap is made up of two lobes. And in the center of the trap is called the mid-rib. So mid-rib usually is actually the center of the leaf, right? So this center of the leaf is called the mid-rib, means middle rib cage, mid-rib, okay? And then at the side of the leaf, right? So the side of the leaf is this one here. It's called the margin, okay? So at the margin of this leaf, specialized leaf, there have got spine or hair. So this is called the spine. So they call it marginal spine or hair, means hair or spine located at the margin of these two lobes, okay? So this whole thing is considered as one leaf, you know, okay? Interesting, with two lobes, huh? So it's one leaf with two lobes, okay? And these are the margins. This is the mid-rib. And the margins has got spines. And then the mid-rib got three hairs, okay? Three hairs. Sorry, not the mid-ribs. The lobe has got three hairs, okay? Now, what about the glands? Very important. There are two important glands on the upper surface of the lobes, okay? So this is for their function. The first one is nectar secreting glands. They are also called alluring glands because they want to lure, means attract the insects to come to them, right? So how to attract? They will secrete nectar, which is food for the insects, right? Okay, like honey, very sweet, okay? So alluring glands. Number two, they also got digestive gland, is to actually digest the insects that have been trapped lah. So the nectar secreting glands or alluring glands, they are located at the outer margins of the lobe, okay? About 1/4 inch from the trigger hairs. That means to say that this is the two lobes, right? Okay, so this is the margin, isn't it? Okay, this is the margin with spines. So they are located at the outer margin of the lobe. Okay, so they will secrete the nectar, right? And all these are located about 1/4 inch from the trigger hair, right? We have got trigger hair here, isn't it? So from here to here is about 1/4 of an inch, right? 1/4 of an inch, right? So why is that 1/4 of an inch is very important, huh? So if the insects come here and feed on the nectar and the insect is very small, right? Okay, let's say this is the lobe, right? And then the insect is very tiny, right? So no matter how it moves around, it will not be able to touch the hair one, you know. So it will not stimulate the hair. But if the insect is slightly bigger, okay, so as it start to feed on the nectar and start to move around, it will actually touch the hair one because it's big. So the size of the insect must be about more than 1/4 inch. So when it feed on the nectar in those areas near the margin of the two lobes, it will by chance it will touch the sensory hair. Okay, so therefore this is for conservation of energy. So it is large enough. So only those insects where the size is more than 1/4, it is large enough for the plant to receive sufficient nutrients from the insect once it traps the insect. Because when it traps the insect, it needs energy. So you must make sure that whatever that touches the sensory hair must be big enough. Then when it starts to use the energy to trap and to digest, they don't waste the energy. Okay, so we don't want to waste energy. So it's an energy conservation structural adaptation, right? The location of the sensory hairs versus the location of the nectar secreting glands, okay? So this is very interesting. So these are the digestive glands located in the center of the lobe. Okay, so these are the two lobes here, right? So at the center here, okay, we have got a lot of digestive glands. So these are the cells that will eventually secrete digestive enzymes to the trapped insects. And these cells inside the cytoplasm, they contain red pigments inside the cell. So these red pigments make the two lobes look red color. So this red color actually will attract insects, just like red flower attract insects, right? So you see the lobes here is very, very red. Okay, in the middle here is very, very red, the two lobes, and the sides got the margin has got spines, and near the margin here there are a lot of these nectar secreting gland, alluring glands, okay, over here. Very interesting. Okay, you can see the three hairs, right? Arranged in triangle location, arranged at the edge of the triangle, okay? So what are the four phases to trap insects, right? First, the lobes is in an open phase, so the picture shows you open phase, then followed by shutting phase, narrowing phase, and closing phase. So there are four phases that will the two lobes will go through, okay, to trap the insects. So the open phase looks like this, okay? So the nectar and red color is in the upper surface in order to attract insects. So nectar will be here, secreted here, and the red color is here. So this will attract the insects. And the marginal spines are those spines here, right? The marginal spines, they are pointing inwards, getting ready to trap the insects, you know, getting ready, okay? Then the upper surface of the lobe is convex. Convex means curve outwards. So you can see curving outwards. So this is called convex because it's V-shaped, right? Upside down V-shaped, so convex. Then the lower surface is concave. You see here, this looks like a cave. So it's called concave. This one is convex because of the pointing outwards, okay? So upper surface is convex, lower surface is concave, right? So why? Because you want to stretch and show the red color, isn't it? You want to show the red color and you want to show the nectar, okay? So this is a shutting phase. So what happens is that if the insects start to move around and feed on the nectar and touches the sensory hair 2 times in about 20 to 35 seconds. So within 20 to 25 seconds, they move around and touches the hair 2 times. So after they touch the sensory hair 2 times, 0.5 second later in the second touch, okay, 0.5 second later in the second touch, then the trap will close already, like the one in the picture, it will close, okay? So the marginal spines will loosely interlock with each other. So it will remain like this, okay? So this forms a prison bar. So it's trapped inside already, the insects. So the trap will remain in this structure for 30 seconds. Oh, sorry, 30 seconds, 30 minutes. Okay, it will remain here for 30 minutes. The reason is if the insect that's trapped happen to be very tiny, the insect will be able to crawl out from the spaces in between the spines, right? Like the prison bar, you got the open space, right? You're smaller than open space of the space between the two bars, you can actually go out. So small insects will go out, and then there's no more, the trap will no longer do any action further. So no need to waste energy, okay, to secrete enzyme and digest, okay? So that's it. So small insects may escape through the gaps between the marginal spine for the plant to conserve energy. So if the insect is too big, right, of course it starts to move around inside the trap there, and as it moves around to look for a place to go out, it will touch the sensory hair again and again, right? So if it continue to touch the trigger hair, right, then the next phase will be initiated already or stimulated. So what is the next phase? The next phase is called the narrowing phase. So you can see from here, which is the shutting phase, right? Can you see this part here? The two lobes, right, near the margin already touch each other. Initially, there's an open space. You can see not there's a space. Now, no more space. They already touch each other. So it's called narrowing space. And it will continue to narrow downwards. Okay? So narrowing phase. So the margin means the edge of the two lobes will continue to close until they are flattened against each other, until a small space remains near the mid-rib. So this is the mid-rib, right? The center of the two lobes. So until a very small space remains near the mid-rib, and this will form the external stomach of the plant. External means the cells will secrete the enzyme out of the body of the plant, and this space here becomes an external stomach to digest the trapped insect. Okay, so this is the last phase called the closing phase. All right, so this is the closing phase. So what happened here is the lobes are completely flattened against each other, and the pressure may actually crush the insect's body. And then the digestive enzyme will be secreted and will drown the insect. Okay, so at this moment, the upper surface is concave. Can you see? The inside is already concave, got cave, and the outside is convex, okay, of the trap. Okay. So each trap can catch prey 3 to 4 times. That means they can close already, right? After digesting the insect, everything absorbed already, they open up again. Then they can trap again another time. So maximum 3 to 4 times only. After that, cannot already. Okay, after that, stop functioning. So I'm going to look at the detailed function of how this whole process happened in detail. Okay, how the trap closes. So we will use stimulus response model again and again, so it's easy to learn the whole process. Okay, so for stimulus, it's the mechanical movement of the insect. Okay, the insect moves and then it touches the sensory spine, right, on the lobes of the trap. Okay, so these trigger hairs are the receptor. So stimulus is the mechanical movement that touches the trigger hair cells. Okay, so that will lead to a transmission of this stimulus energy in the form of electrical energy, means action potential. Okay, transmission to effector directly. So the effector are the mid-rib cell. Mid-rib cell means the cell in the center of the two lobes. Okay. Okay, so the mid-rib cells all along here. Okay, so, and also secretory glands, which are located here, right? The secretory glands. So these are the effectors. Okay, so first, mid-rib cell, the response is the trap will close. So mid-rib cell will be activated to cause the entire two lobes to close together. Okay, and then after closure, then secretory glands will be activated to secrete digestive enzymes to digest the insect. Okay, now we're going to look at the detail. This is very interesting. The detail how After stimulus, action potential is generated and transmitted along the lobe all the way to the cells at the mid-rib of the lobe, right? Okay, that means this is the lobe. This is, let's say, these are the hairs. Okay. So starting from here, how the action potential is transmitted all the way until the mid-rib of the lobe. From one cell to the next cell to the next cell, all the way to the mid-rib. So we're going to look at it now, okay? So step one, touching of the same or different trigger hair. So it can be touching the same hair or different hair because there are altogether 6 hairs there, okay? So touching the same or different hair 2 times in succession within 30 seconds, plus minus, will stimulate the formation of action potential at the membrane of the trigger hair, okay? Like the receptor cells, okay? Like our receptor cell. So this stimulus will cause the cell surface membrane to depolarize, okay? So touching of this one by the insect will cause the membrane to depolarize. Very similar to our neuron, right? Our finger touches the pin, right? Then the tip of the nerve cell depolarize. So now the membrane at the tip of the sensory hair depolarizes. Now, for plant, they use chloride ions. They don't use sodium ions here. So what happened is that chloride ion diffuse out of the cell. So chloride ion efflux. Efflux means exit. Exit means to go out of the cell. So as the negative charge goes out of the cell, inside the cell become more and more positive. So the membrane potential will increase. Okay, same as in our animal neuron, sodium ion comes in, then positive, inside becomes more positive. Chloride goes out, inside also become more positive. Okay, so membrane depolarizes. So cancel off this one, right. So if the membrane potential is above the threshold potential, action potential will be generated. So very similar, right? Okay, action potential. So once the membrane potential goes above the threshold, then eventually action potential is generated. Okay. Now, action potential will then travel along the cell surface membrane. Okay, like this. So can you see the outermost region here is the cell wall? So this inner black line is the membrane. So I'm showing you the red arrow is the action potential traveling along the membrane all the way. And then it can travel from cell to cell through the membrane at the plasmodesmata, because the cell surface membrane of 2 neighboring cells are connected at the plasmodesmata. So that's how they send the action potential from cell to cell to cell to cell, all the way from the sensory hair here to the cell at the mid-rib, okay, of the trap. So then when it arrives to mid-rib, right, how does it cause the trap to close? So action potential arrives at the effector. So the effector is the mid-rib cell, because it is the mid-rib cell that changes in structure that cause the 2 lobes to close, okay? So therefore the effector is the mid-rib cells, which we also call the hinge cell, right? Why do we call it hinge? Hinge is like, you know, the door, right? Your door has got hinge here. That's why your door can open and can close, right? So these 2 flaps here of the 2 lobes, right? Okay. So this part is also like the hinge of the door. It allows these 2 flaps here, or the 2 lobes here, to go this way and that way. So this one becomes the hinge. So they call the cell there called hinge cell. Right? The change in size of the hinge cell is the one that can cause the 2 lobes to close. Okay, that means the hinge cell originally is this size. Then after activated, it becomes this size. Okay, bigger. Okay, so when it becomes bigger, it stretches, and then the rest of them don't stretch, so they will close. Okay, how to illustrate? Okay, it should be like this. The cell original is like this. After that, when it stretches, it becomes like this. So original, the shape is like this, right? After stretches, the shape is like this. Okay, close. right? Okay. Now, how do I erase this thing? Okay, auxin. So you see, so action potential travel all the way from sensory cell and then goes to the hinge cell already, or the midrib cell. So at the midrib cell, once action potential arrives, it will cause a plant growth regulator, auxin, to be secreted. Okay, so this auxin will increase the activity of proton pumps. Okay, so you can see that this is a midrib cell here, or hinge cell, and at the membrane of the cell, we got a lot of this proton pump. So once auxin arrives, okay, number one, action potential arrives. Number two, auxin secreted. Number three, auxin will stimulate proton pump to pump proton out from the cell. So where is a proton pump to? So this is the cytoplasm of the cell. Can you see? So the proton is pumped across the membrane of the cell, and it comes to the cell wall. There are a lot of spaces in the cell wall because cell wall is made up of cellulose. Where's my cellulose? Cellulose, okay? The orangey rod shape is the cellulose, and then hemicellulose, the smaller black color chain-like structure, and then pectin, the green color structure, okay? Hemicellulose and pectin will hold all the cellulose together. All right, so there are a lot of spaces in the cell wall. So proton ions are being pumped into the spaces in the cell wall. Okay, so proton pump pumps proton out of the hinge cells into the cell wall spaces, where cell walls are made of cellulose, hemicellulose, and glued together by pectin. So they call it calcium pectate, the pectin. Okay, so because of the H+ ions in the cell wall, this will actually reduce the pH or increase the acidity, okay? So this one in turn will activate a protein called expansin, number 4, okay? So this acidity or this decrease in pH activate a protein called expansin. So if you look at this word, expansin is to cause something to expand, something to increase in length and size, right? So what does it do is it helps the cell wall to be able to expand. Now the cell wall cannot expand because all the cellulose are held together by hemicellulose and pectin. So when expansin come in, how does it help the cell wall to expand is it will break down the non-covalent bonds between the cellulose microfibrils and the hemicellulose. So it will break the non-covalent bond between cellulose and hemicellulose, and it will also digest the pectin away, which glue the cellulose together. So they cut away all the bonds between cellulose, hemicellulose, and pectin. So they are no longer held together. So the cellulose fibers are no longer held together. So the moment they are not held together, they are free to move, you know. Okay, they're free to move already. So now the microfibrils, cellulose microfibrils, they are able to move past each other easily. So the cell wall becomes very loose. So the moment water enters the cell can expand continuously. Okay, because the cellulose wall is no longer rigid already. So that's why it's activated by expansin. Okay, causing the cell to be able to expand by breaking all the bonds between cellulose, hemicellulose, and pectin, so that the cell wall can stretch because the microfibrils are free to move already, move past each other. Okay, so once the expansin has already digested and the connection, the bonds between cellulose, hemicellulose, pectin, step number 5 is calcium ion will then stream into the cell. Okay, why? Very easy to understand. Because H+ are the positive charges already go into cell wall, right? The H+ which carry positive charge already go out of the cell. You have to replace this positive charge. So they use calcium to replace this positive charge. So calcium ion streams into the cell, and then you know calcium is a solute. It will reduce the water potential of the cell. So because of that, water, okay, water will then streams into the cell by osmosis, right? So therefore, the hinged cell will swell, okay? So it will swell in a certain way that will cause the two flaps to come close to each other and close, okay? So because of swelling, it should not swell in the same length, right? It should swell in a triangular form, right? One side will stretch more, the other side will stretch less, okay? So then the two lobes, okay, so the midrib cell. So it will swell in such a way that it becomes like this. I think wrong already. It becomes like this, okay? Then the rest can actually close. Okay, you stretch some more, it will, and then it will close some more, okay? Okay. So how the trap closes, step one, stimulus, there's a mechanical, due to mechanical movement of the insect that touches the receptor, which is the trigger hair cells or sensory cells, and that will cause action potential to be transmitted from cell to cell, okay, from the trigger hair cell along the rest of the cell until they reach the effector. Effector is the midrib cells. Okay, we look at midrib cell first, and then the response is the trap closes. Okay, now after the trap closes, right, if the insects continue to move inside the trap and keep on triggering the sensory hairs, it will cause the two flaps or the lobes to close tighter and tighter, right? And at the same time, that means to say that it keep on sending action potential to the midrib cell keep on expanding, so this one keep on coming closer from like this, become closer and closer and closer and closer, okay? Then it also stimulate the digestive gland to secrete enzymes, okay? So continuous stimulation of trigger hair sends more action potential, not only to midrib cell to make this part here continue to close, but also to activate digestive gland cell to secrete secrete enzymes. So the way the enzymes are secreted, right, is very similar to our synaptic knot. Okay, so action potential arrive to the digestive gland cells, right? Calcium ion streams into the cell because the calcium ion channels open. Then secretory vesicles containing the digestive enzyme, now are not acetylcholine, will then move towards the membrane, fuses with it, okay, move, one mark, fuse, one mark, and then release the content by exocytosis, one mark, okay. Then the digestive enzyme will flood and drown the insects and start to digest the softer part of the insect. That means the exoskeleton, right, the hard skeleton outside insects cannot be digested. Okay, the internal organs, internal flesh, everything can be digested. Okay, so this is after complete digestion, the trap will open back, and this is what you see. Whatever remain is actually the exoskeleton, means external skeleton of the insect. The internal juicy part already digested and absorbed. Okay, left outside dry exoskeleton. Okay, so the trap remains shut for 5 days for digestion to happen. Okay, why 5 days? Because it is an external environment temperature, so it is a lower temperature, so the rate of digestion is slower compared to if, like, let's say in our stomach, temperature is 36.7, okay, so it's of course faster, okay. So nutrients absorbed into the plant cells. Once the insect is digested, the cells on the upper surface of midrib grow slowly so that the leaf opens up and the tension builds in the cell walls of the midrib. Okay, so that means after some time, the cells on the upper surface will continue to grow bigger and bigger so that the leaf will expand and open up again. Okay, so the hard chitinous exoskeleton, right, is made of chitin, okay, remains or is washed away by the rain or blown away by the wind. Okay, so overall, how does the trap closes? Recap again, okay? There's a stimulus where the insect's movement touches the receptor, and the receptor is our trigger hair cells or sensory hair cells. Then action potential is generated by chloride moving out of the cell, right? Membrane depolarized until above threshold potential, action potential generated, pass from cell to cell through plasmodesmata along the membrane, reaches to the effector, which is the midrib cell. Then inside the midrib cell, a lot of things will happen, right? Auxin will be produced, proton will be pumped out of the cell. Proton is being moved out of the cell, okay? And then the acidity will cause expansin to actually break the bonds between cellulose, hemicellulose, pectin, okay? Then after that, calcium ion will move into the cell, water potential of the cell increases, water enters by osmosis, the midrib cell will swell, and that will cause the trap to close. Then secretory glands secrete the digestive enzymes to digest the insects. Okay, all right. So there's still more time. Hang on. So therefore, I'll take